Hey, so I'm here with Ian Lipkin to discuss the new study that was just published. And so, Ian, why don't you first put it sort of in the context of the work that your group has done and, where this is in sort of that overall context um, from when the group was starting to work on the issues.
Historically, I can go back to really the mid 1990s
Hmm.
when when I was asked by Brian Mahi to look into some reports that a virus called Bornavirus was responsible for ME/CFS. And at that time, we used a variety of genetic tests, antibody-based tests, and we did not find any evidence for this particular virus, despite the fact that there had been a lot of enthusiasm for it in Europe and in Japan. But what we did notice
I mean,
was that
as a possible source of ME or
positive cause
trigger
of
for
disease.
ME/CFS.
Yeah.
So,
So, but what we did find was that three-quarters of the patients we studied had high levels of antibodies that were directed against something wasn't seen in nature. I'm not saying that means that it was a stealth virus or anything of that nature. It was simply that they had polyclonal B cell activation. So they had exaggerated immune responses. So when I reported this, I said, we don't find any evidence for this virus Borna, but we do find evidence that there's an objective problem. And we really didn't do much more with it at that point because there were no resources until um, a donor to New York Presbyterian Hospital came to me and said, my son has this disease. Can you help him? Can we look to see if there's an explanation? So working with an infectious disease physician, we took peripheral blood mononuclear cells and we found high levels of Epstein-Barr virus.
mhm.
He was treated for that infection and he improved. And he went from, barely making it through school to graduating toward the top of his class. Father was very grateful and said to me, everybody should have access to these kinds of diagnostics and improvements and so forth. And I'm going to help you get started. Tell me what is it you need. I said, well, first, we need a biobank because there are many people who could look into samples if we had them and figure out whether or not there are certain markers for disease that could give us insights into why people are sick and what we can do about it. So this goes back really to, the mid 2000s. And we got started and we found these cytokine abnormalities in spinal fluid and in blood, and slowly began to build the case that this was a biological illness. And remember, at this time, there were still a lot of people who were thinking of this as primarily psychological.
Hmm. Yes, I remember well.
Right. So this was an epiphany for some people in the field, but it began to slowly build some sort of momentum, and we began looking at all sorts of things. We put together the Center for Solutions for ME/CFS, and I got people who were experts in RNA biology and metabolomics and proteomics, and we slowly began to put together, a story that began to become cohesive. And there were definitely protein abnormalities and metabolomic abnormalities and so forth. So we had just enough in the way of resources to begin to do with some support from this philanthropist, looking at post-exertional malaise
Okay.
because we had to focus somewhere. So we put people through exercise testing. We did cognitive testing thereafter. We collected blood prior to exercise, after exercise, 24 hours after exercise. And we began studying the metabolites that were there as an index to what was happening inside of cells, proteins that might be turned on or turned off, cytokines were expressed, and so on. And what was the most striking to me was not the metabolic abnormalities, although they pointed to damage to mitochondria and peroxisomes. But the most exciting thing, because I thought it was potentially actionable, was that if you looked at these assays we did that were called true culture assays, which is where you take peripheral blood from an individual before and after exercise, and you expose the white cells that you'd collected to stimuli of innate immunity, things like staph enterotoxin B, or which is a superantigen, or polyinnocene cytosine, which mimics a virus, or heat-killed Candida antigen, or lipopolysaccharide, which mimics a gram-negative bacterium.
Just could you define superantigen for some of those out there who wouldn't know what a superantigen is?
A superantigen is something which directly stimulates inflammatory cells. It doesn't have to go through a series of intricate pathways. It's a direct effect.
Okay.
And so, as a consequence, what you found was that the people with ME/CFS had stronger responses to all of these stimuli than their healthy age sex match controls. And the responses were higher in women than in men, and higher in women over the age of 45 than women under the age of 45. And there's a correlation between levels of estradiol,
Hmm.
in women over 45 and under and so forth, and the extent of the response. Now, the initial kits that we used were fixed in the amount of stimulus that the blood cells saw, but by using a sort of a dose response curve, different levels of these compounds that stimulate immune cells, or the length of duration of exposure and so forth, we began to tease at even more dramatic effects. Now, about a year earlier, we had published our first large
And
microbiome.
just for this, you're talking about the results that you just published. And
Yes,
okay.
these results just published. But now I'm going to show you how this dovetails with earlier work that we've done.
Hmm.
So we published this large microbiome paper um, about a year earlier, in which we showed that people with ME/CFS had a very different fecal microbiome than normal people,
Hmm.
healthy people, I should say. They had fewer bacteria that produce short-chain fatty acids, one in particular called butyrate. Butyrate is very important because it regulates immune responses, it suppresses them, and it also maintains the integrity of the lining of the GI tract.
Hmm.
So putting two and two together, we thought about this and said, if the lining of the GI tract is more permeable, and if this regulatory molecule associated with these bacteria called butyrate, short-chain fatty acids, is reduced, and we have trafficking of material from the inside of the bowel into the systemic circulation, this in somebody who's prone to a hyper responsiveness to these stimuli could have, a flu-like syndrome, right? Feeling ill, cognitive dysfunction, post-exertional malaise, all of these things. So we're still not completely confident the model because we really would like to have an animal in which we could study this. But it's beginning to give us a sense that there is damage that is done to these immune cells, and there's damage to cells that produce these organelles inside cells in the body, in the brain, and elsewhere that results in dysfunction. And you can tie all this together by thinking about abnormalities in the GI tract, these regulatory molecules that control inflammation, and the exaggerated inflammatory responses that we see in individuals who are afflicted with ME/CFS. So how would this translate into something that would be useful? Well, if we can find out where these pathways go awry, we may be able to repurpose drugs. Typically, drugs are used for treatment of cancer or other autoimmune disorders that might be useful in mitigating symptoms of ME/CFS
Hmm.
or other post-inflammatory disorders like long COVID or post-treatment Lyme disease, any one of a number of of disorders where you have similar sorts of phenomena. So
Um,
that's, sorry, go on, go ahead.
Question. Yeah. So that you were saying that's where we are more or less. I guess with this. So one of my questions is, you've
Well,
again.
we're a little further. We're a little further. I want to throw out a couple of other interesting
Okay, you do that, and then I'll ask my question.
coming attractions. So um, the guys who led the work on the microbiome were Brent Williams and Cheng Guo. Cheng has gone back to China. Brent is, I think, in Oklahoma at this point. I don't really know. Unfortunately, when he lost his funding, we lost him. But they're the ones who did that original work on the microbiome. The work that's been done now with, with these inflammatory cells, the lead authors on this, the two very, very talented junior people were Jason Shea and Ahmed Ranjan. Jason's left for China again, related to loss of funding. Ahmed is still with us.
Hmm.
Fortunately, been able to retain Ahmed. So, and we're going to continue to work on trying to understand triggers of innate immunity. Now, one of the things that we need to do, I think, is to try to figure out what might trigger the damage in the first place. So we have three projects that are funded that will allow us to approach this in a systematic way. The first project we call the Good Day, Bad Day Project, really named that by Tony Komarov, a name that many of your viewers will know. And the idea here is that on days when people feel particularly poorly or particularly good, we can send somebody out to collect samples, and then we can look 2, 3 weeks later and ask whether or not the proteins, the metabolites, the viruses that are present, in saliva might differ, right, as a trigger. We can also look at whether or not antibody profiles directed against bacteria or viruses differ. So,
Hmm.
for example, if I think that Epstein-Barr virus might be implicated, but I don't find Epstein-Barr virus in blood, that doesn't mean it's not in saliva. So we could track it in this way. And 2 to 3 weeks after we do that first collection, we can go back and collect blood. And if in fact there was an intercurrent infection with Epstein-Barr virus, we would then be able to detect antibodies, not only antibodies directed against the virus, because 80% of the population has been infected with EBV. That's not going to be terribly informative. But there are specific proteins that are made by the virus when it reactivates.
Hmm.
And if you can show that those antibodies go up, indicating a reactivation, that can be really, really helpful.
I mean, a
It can tell you
reactivation
some,
as a reactivation where there's ongoing replication of.
right. So most of the time, these viruses are dormant.
Mhm.
They're not active at all. But if they're reactivated by something, you'll see you may not be able to detect the virus, but you can detect immune responses to it. And unlike the other serological approaches many people use, ours are very, very narrow. So we can look at the entire proteome of the virus and specifically focus on those genes that are turned on when the virus reactivates.
Mhm.
So those are the sorts of things we're doing. That's work led by Nisha and Misha. So that's sort of project one. Then we started a really interesting project. Originally, we were going to do this work with Chris Ponting
Mhm.
in Edinburgh, who headed up this decode project, but we were told that we couldn't work outside the continental United States. So what we did is we substituted a very interesting project with the University of Utah and the Bateman Horne Center.
Okay.
And the idea there is we're looking at pedigrees, families where there's more than one person affected with ME/CFS. And we can look at similarities in genotype using whole genome sequencing. So we can find, instances where people have multiple copies of a gene that's implicated, or there's a problem with the regulation of that gene. something which is upstream of the gene or downstream of the gene, as well as the gene itself.
And
So
just
that
to clarify, that's
insights.
a replacement. That's a replacement or a sort of substitute for the project that was announced a couple of weeks ago originally, or that seemed to be in the
Yeah,
works.
the original GWAS project was
Was
called the Genome-Wide Associated Study
right.
that we were told we couldn't do.
Okay.
So you make lemonade. And in this case, I think
And this is
whatever you
told
would have
by
asked,
told by who? Just told by is the federal government making these kinds of things, or
I
told by?
don't want to wade into that. It's
Okay.
not helpful. And I've only just gotten my funding back, David. So
Okay.
just
I
leave,
understand. I understand. I
let's
understand.
leave that sleeping dog lie.
Okay.
Though frankly, I would think that they would love the kind of work we're doing because it's going to give us insights into chronic illness.
Well, hard to
Anyway,
understand. So anyway, that's the second project. Then the third project you said that you had.
that's the second project. The third project is also fascinating. So many people aren't aware of this, but since the mid 1980s, the Department of Defense has been collecting blood from people annually to get some sort of sense of how many people have HIV infections in the Department of Defense, across the whole Department of Defense.
Mhm.
So everybody's active duty has blood drawn. So we can go back and see who has reported any disease, chronic fatigue syndrome, unspecified, um, ME/CFS using ICD-10 codes, whatever the case may be. We get as close as we can. We use AI. We try to find people who, using the old criteria, still would have met, what we now think of as ME/CFS. And we can get blood that was collected a year before, 2 years before, blood collected the year that they're diagnosed, and the year after that. So you can actually see whether or not there is some trigger, a viral trigger, a
Mhm.
bacterial trigger. You can do metabolomic studies and see whether or not there are any sort of biomarkers which would have indicated that somebody was on their way to developing disease. So all of these things get us, they're not, they are intellectually interesting, but that is not our motivation. Our
Right.
motivation is to understand the underpinnings of disease so we can, develop hypotheses about what might be useful to prevent or treat disease.
So going back to my question that from before, in the study, you talked about multiple different processes that you've identified through various the differences pre and post exercise, and then through the stimulation, in the of the cells. Is it your sense that all of those things are all going on, or that most of those things are going on with most of patients? Is there a unified thing that includes all of the things you see, or is it that everybody's got something somewhat different? So collectively, these are the things that we see, if you understand the distinction I'm trying to make there.
I think I understand. I'll try. So, I believe in this concept of Occam's razor. The simplest explanation is the one that's likely to be true. So, what I like about this innate immune response abnormality is that, first of all, it ties in nicely with the data I was talking about with respect to dysbiosis in the intestine, permeability of the intestines, bacterial products floating into the systemic circulation, not being dampened, and the response, therefore, initiating this whole sort of sickness behavior, which results in, people feeling the way they do. Um, but there are all sorts of components to this. One of the things that comes across in the paper is that we find abnormalities in the way in which certain neurotransmitter pathways function.
Right.
You actually looks like you're
Tryptophan.
producing, you're producing quinolinic acid, things like that are toxic. So I cannot yet come up with a single, primary problem, but I do think that the initiation for this stuff is in people who are genetically predisposed to having abnormal immune responses, that there's a trigger which sets off, a cascade of events that are intracellular and that should be actionable. more we understand about how these pathways are impacted, the better we're going to be in terms of sorting this out. I make the analogy to cancer. You know, you and I are pretty much the same vintage. In the old days, what do we used to do to treat people with cancer? We would use these shotgun approaches. We would kill cells, and the idea being that the cells that were replicating the most rapidly were the cancer cells. So we would try to keep them at bay, realizing at the same time we were causing toxicity for a whole range of other cell types. Same principle with radiation. More recently, we have this bespoke approach, right?
Mhm.
Precision medicine. So our goal is to try to get to precision medicine with ME/CFS. And I think we're going to get there by using various tools that can manipulate immune responses and can interfere with certain aspects of cell cycle. Is it going to be complicated? Yes. Is it something that's 5 years away? I think it's going to be less than that. I think we're getting close.
Um,
Um, the reason everything is so slow is that there are no resources. I mean, look, we lost 2 years. I mean, you know, the first time we sent our application in, it went to a study section where people said, I don't understand how this antibody array is going to help me understand what's happening with ribosomal RNAs. And I said, It doesn't. These are completely, they're completely separate pathways. They had people reviewing this who didn't know what they were doing. So we had to come back around again. We lost 9 months with that. We lost time during the pandemic. And then, as you know, Columbia, lost all of its funding.
right.
So, and even when it came back, David, it was slow. So, um,
So
thank God
there,
for
just to
philanthropy.
finish on that, you have the funding from this that had been written about from the center has returned slowly, even
It
though
has
there's been large
returned
funding.
slowly, and what we have is a no-cost extension against which we are spending to keep things going. And then, of course, in the period of, of funding loss, you lose people.
Right. I wanted to
So
ask,
you have to re-recruit.
what is the current state of evidence for chronic inflammation? And also, I linked that to the previous study you did where you found distinctions in those who had been sick for a shorter time, who seemed to have more pro-inflammatory cytokines happening, and those who were sick for a longer time, who seemed to have a different phenomenon happening with the immune system. So I'm just wondering, overall, in your this study suggests, chronic inflammation or processes that indicate chronic inflammation. So where is that?
That doesn't mean that at any given point, I mean, if you look randomly, if you collect bloods randomly, you may not see anything. And you may have inflammation in tissues
Mhm.
that you don't necessarily see in blood. But there's no question but that there's a that the threshold for triggering inflammation, be with ME/CFS, is lower than
Okay.
the threshold in people who are healthy. So anything can set this off. When you brush your teeth, you have bacteremia. If you have a bowel movement and you're constipated, you're going to have bacteremia. If you have, if you have reactivation of Epstein-Barr virus in your oropharyngeal, mucosa, you're going to have inflammation. So all of these things can give rise to, the abnormalities that we see, not only in terms of inflammation, but we see problems with the Krebs cycle, which generates ATP, and so there are a whole range of things that are, um, that occur as a result of this reduced set point, right?
And what does all this do to the still ongoing discussion about whether this is a functional neurological whether a functional neurological disorder or just a functional disorder writ large, and with sort of mysterious processes that we don't really know what are going on.
Well, the problem with the term functional is that everything is functional or dysfunctional. But what most people mean by functional when the is something disparaging. They're claiming that it's something that you can control, right? They're saying that it's primarily a psychological problem. Do I think that psychology plays a role in virtually everything? Yes, certainly. And it plays a response, it's important thinking about, which people recover better from, heart attacks or strokes or anything else. But that doesn't mean that the primary lesion
Hmm.
is psychological. It isn't. I mean, there's no question but that the data we have over years, and other people have the same data, indicates that this is a cellular biological phenomenon. And we think that the majority of the damage is related to immune system dysfunction. And
And
our work here clearly shows that the immune system is dysregulated. And if you tie that together with the dysbiosis and all the other things, you can begin to chip away at how you might make people feel better. So there are people who are going to have dysbiosis. Not everybody has it, but everybody has it, should be treated, with probiotics and prebiotics. There are people who have recurrent herpes virus infections. They should be treated. Now, we don't have great drugs for the herpes viruses. I'm talking about the EBV.
right.
We need better ones. There are people who have abnormalities in innate immune responses. These are things that we think we can begin to repurpose drugs.
Where
in
does the
as
orthostatic
well.
intolerance fit in with this?
Right.
Hmm? Where does the orthostatic intolerance part of it fit in with this?
So I have not focused on orthostatic intolerance. I could tell you that I think it may have something to do with the autonomic nervous system. I mean, but that's tautological. It's,
Okay,
you know,
so
um,
that's not let's move on. Um, any final things you wanted to say about the study that I haven't asked?
just that there's a whole panoply of abnormalities, right? Having to do with energy metabolism, mitochondria, peroxisomes. There's a lot, there's a lot to tackle. And what we need to do, I think, um, is complete the three studies that I'm talking about, which I think we can do fairly rapidly, um, which will give us more clues. And during the interim, one of the things I'm very excited about is, there's a paper that just came out in PNAS where people looked into who had reported what had been useful and helpful and what had not been helpful. I think we need to do more of that sort of thing because,
PNAS, just to be clear, Proceedings of the National Academy of Sciences.
right, yeah. So, I mean, we don't have the resources that were allocated for the long COVID program, for example. Um,
And let me just ask that. We didn't discuss this. How do you place long COVID, or I'd say long COVID with the ME kind of subset of long COVID, and maybe more all of long COVID, I don't know, in this framework or within this hypothesis.
well, I think the most important, you know, outcome for patients with ME/CFS is that as a result of long COVID, people realize that you can develop a chronic debilitating illness with many features of ME/CFS after an infection. It has given it a legitimacy. That said, I prefer to focus on ME/CFS. There are many people doing long COVID research, very few doing ME/CFS research. And the efforts, that I and others have had to try to suggest that patients with ME/CFS might be a good control group for long COVID, for example. So we could get people into clinical trials have been unsuccessful.
Okay.
So what we do in our work is we focus on people who have ME/CFS prior to 2020 so that it's not confounded
Okay. With the long
by
COVID.
by long COVID. I mean, the largest long COVID trials are people being treated with Paxlovid. That's not going to help anybody with ME/CFS.
And your cohort are at both you use both the Fukuda and you use the Canadian consensus criteria. And do they all pretty much meet both, or is there any
Yes,
differentiation? Yes, they all meet both.
do.
Okay, so they all have
So
post-exam.
we try to be as stringent as we can.
Okay. Um, okay. Thanks, Ian. I appreciate it.
My pleasure. Good luck with your roof.